number 55, which is the first marker, antigen or antibody, which will become positive after exposure to hepatitis B. Choices are HBs antigen, B, anti-HBs, C, anti-HBe, D, IgG anti-HBc. So, understanding the hepatitis B blood testing is very important in analyzing the interpretation, what is the stage, what is the current condition or status of your hepatitis B patient, or in case a normal person. Okay? So in this case daw, ang tanong daw, after exposure ng hepatitis B. Okay, in hepatitis B. Okay, so from our previous discussions, okay, hepatitis B surface antigen, okay, is the first serologic marker to appear, okay, in a new or acute infection. Okay, first key marker to appear in a new acute infection. Okay, this can be detected. This can be detected in earliest possible time at 2 to 12 weeks post-exposure. Okay, post-exposure. Okay, this is during the acute stage, okay, of the infection. And this will become undetectable in 12 to 20 weeks, okay, after the development of anti-HBs antigen. Okay, after the development of anti-HBs antigen. So, it will be detected, okay, 2 to 12 weeks post-exposure and be undetected, okay, in 12 to 20 weeks. Okay, this is due to the development of antibody against the HBs antigen. So, here in the illustration from Turgeon of hepatitis B infection, you can see here that the first marker is first marker present is the HBs antigen. Okay, it is the HBs antigen. So it can be detected, okay, 2 to 12 weeks, okay, after exposure. Okay, this is followed by the HBe antigen, okay, but it will later on decline, okay, before the disappearance of HBe antigen. Okay, before the disappearance of the HBs antigen, mawawala din po si HBe antigen. Okay? Very important din po na malaman, no, na the first antibody to appear here, okay, is the anti-HBc. In particular, the IgM anti-HBc. Okay? It will persist for about 6 months of the infection. And we have here the anti-HBe. Okay? This is positive at the end. Okay? It is positive at the end of the acute stage of the disease. And then we have your anti-HBs that will appear once the HBs antigen is undetected in the blood. Okay? Once undetected in the blood. Okay? So here are the important diagnostic tests, okay, per stages of the hepatitis infection. We have the incubation period. HBs antigen is, okay, detected. In a prodrome acute disease, okay, there could be a presence of HBs antigen and anti-HBc. Okay, and then in the early convalescent stage of hepatitis B infection, we can observe the presence of anti-HBc, the onset of anti-HBc. Okay, and then in the late late convalescent stage, we can observe anti-HBs and anti-HBc. In this case, yung anti-HBc na makikita natin dito, Okay, this would be the IgG, okay, anti-HBC. Okay, so 55 po, first marker to be observed. This is the HBs antigen. Okay, so A is the correct answer. 56, the presence of HBs antigen and anti-HBC and often HBe antigen is a characteristic of what phase? Here, present po si HBs antigen, si anti-HBC, tsaka si HBe antigen. And this marker suggests the early acute phase of HBV hepatitis. Okay, ma'am, bakit hindi siya recovery phase of acute HBV hepatitis? Remember that your HBe antigen, okay, it appears after HBs antigen. Okay, it appears after the presence or the detection of the HBs antigen. Okay, and in recovering patient, those who start to recover from the infection, HBe, antigen disappears. Okay, HBe antigen disappears. So you can see here, that the HBe antigen is still present, especially at the early acute phase of the disease, and later on it will be undetected. It will be undetected. So early convalescence phase of the HBV infection, this is helpfully tested, or detected using the total anti-HBC. The total anti-HBC. Why? Because at this stage, if naalala niyo yung kaninang graph, at this point, anti-HBC is at peak. Yung relative concentration ng anti-HBC, is at peak, indicating that the phase is at early convalescence phase. We shall remember that anti-HBC is the first marker to be detected in the patient blood, in the first antibodies, specifically si IgM anti-HBC. Si IgM anti-HBC. Useful for detecting HBV infection when HBs antigen is no longer detectable. Indicating a window period. Also this marker, this is used to screen blood donors. Ano naman yung IgG anti-HBC? The IgG anti-HBC, so this remains positive indefinitely as a marker of past infection. So this is helpfully indicated by anti-HBC. Particularly the IgG type. So number 56, letter A po. We have 57. Which of the following correctly describe primary syphilis? Primary syphilis. So A, stage of syphilis with no sign or symptoms but non-treponemal and treponemal serologic tests are positive. B, T. pallidum enters the body, it reaches the bloodstream, and is disseminated to all organs. C, there is a painless lesion or chancre which appears between 2 to 3 weeks after initial infection. And letter D, we have appearance of skin rash, low-grade fever, weight loss, malaise, and lymphadenopathy. Remember, we have different stages of syphilis. We have the incubation phase, wherein the T. pallidums or the pathogen for syphilis, the agent of syphilis, enters the body and it reaches the bloodstream. And this is the start where the agent is disseminated in the body parts or other organs. So this is the asymptomatic phase, that lasts for about 10 days to 10 weeks. And we have primary syphilis. Okay, primary syphilis, okay, what we see here is the initial lesion, okay, which is painless, non-bleeding ulcer that we call as the chancre. Okay, so painless lesion, okay, or we term as your chancre. Okay, this usually appears, okay, on the average of 2 to 3 weeks after the initial infection. Okay, so the answer for 57 is letter C. Okay, letter C. So, syempre meron tayong incubation stage, sorry. Stage or period. This will be followed by the primary syphilis. We have the secondary. We have latent stage. And we have the tertiary stage. So, sa primary syphilis natin, ano nakikita? We have a chancre. Painless. Okay? Initial lesion, painless, non-bleeding chancre within 2 to 3 weeks. Okay? So, within 1 week after the chancre appears, what we can observe is the enlargement of the lymph nodes. Okay? So, that is the primary syphilis. Okay? So here, in primary syphilis, what is helpful assay? So we can use a dark field analysis of lesions or of this chancre to demonstrate the presence of spirochetes. Okay, the presence of spirochetes. We have the secondary syphilis. Okay, sa secondary syphilis, okay, ang tinatandaan ko dyan, symptoms. Okay. So what we can see here, okay, the presence of skin rashes, low-grade fever, okay, malaise, pharyngitis, okay, weight loss, arthralgia, and lymphadenopathy are the combinations of symptoms that are observed during secondary syphilis. And these symptoms will last for about 4 to 6 weeks. Okay, 4 to 6 weeks. Okay, so during this stage, we can also observe the presence of the spirochetes. Okay, the ulcers develop, okay, on mucous membranes of the bodies, and then we can use serological testing here. So serological tests, okay, are positive during this stage. Okay, so we have po latent stage. Okay, the latent stage of the syphilis infection. So the stage of syphilis shows no signs or symptoms of the infection. Okay, but we can see here, okay, we can see here in latent stage, okay, we have the non-treponemal, okay, non-treponemal and treponemal serologic tests are positive. Okay, these are positive. Okay. So, early latency of the infection, okay, which appears in 1 of 4 individuals with syphilis infection, okay, relapses back to secondary, okay, to secondary syphilis. Okay, so itong latent stage na to, okay, pwede siyang mag-undergo ng early latency, okay, which is observed in 25% of patients with syphilis, okay, and this relapses into secondary syphilis. Okay, we have the late latency, okay, late latency, okay, in this stage, okay, the patient is resistant to reinfection and to relapses. Okay, the patient is, okay, resistant, okay, to reinfection and to relapses. Ano naman po yung tertiary syphilis? Okay, tertiary syphilis, okay, usually appear, okay, or occur 2 to 40 years, okay, of infection. Okay, so after the initial infection. Okay, so what we can see here, okay, is the gumas. Okay, gumas. Ano yung gumas? Okay, yung gumas po, okay, these are the syphilis lesions due to hypersensitivity to treponemal antigens. And these are found, okay, all throughout the body of the infected person. Okay, and what are the other presentation of patient with tertiary syphilis? Okay, meron tayong cardiac, okay, involvement. Okay, they have the inflammation of the aorta. Okay, or we call as syphilitic aortitis. We have aortic valve insufficiency and thoracic aneurysm. These are the possible conditions that will appear in a tertiary syphilis stage. Okay, and also, okay, in a tertiary syphilis, this can be suggested, okay, in case of neurosyphilis. Okay, neurosyphilis class, okay, it can cause blindness and senility. Okay, so take note also the tertiary syphilis, although it appears, okay, beyond years, 2 to 40 years, this phase is not usually infectious. Okay, so number 57 po, the answer is painless lesion or chancre appearing within 2 to 3 weeks after initial infection. Okay, so number 58. What is most likely interpretation of the following serologic results? This is the laboratory test result: RPR reactive, VDRL reactive, MHA-TP non-reactive. Okay, so we have RPR and VDRL as the non-treponemal test or assays, and we have the MHA-TP, okay, as our, okay, treponemal test. Okay, here we can see reactive siya sa mga non-treponemal testing and shows non-reactivity in the treponemal test. Okay, so the suggestion should be A, neurosyphilis; B, secondary syphilis; C, syphilis that has been successfully treated; or letter D, biologically false positive infection. So the positive reaction with the non-treponemal antigen and the negative reaction with a treponemal antigen is most likely caused by a biological, okay, false positive non-treponemal test. Number 58, the answer is letter D, biological false positive, okay, testing. So here, we have, let's discuss the diagnostic test for syphilis. So we can use CSF, umbilical cord, or skin, or mucous membrane lesions for the diagnosis of syphilis. Okay, so we can use dark field microscopy, fluorescent microscopy. Kapag ka dark field microscopy, we use silver stain in staining the lesions, okay, which contains spirochetes. No? So we can also use fluorescent microscopy. Okay, we use the direct fluorescent antibody T pallidum or DFA-TP. Okay, wherein we use fluorescence labeled antibody, okay, that will be directed against the T pallidum lesions. Okay, so serological tests, okay, we can use serological tests in detecting non-specific antibody reagin. Okay, so we use the non-treponemal tests such as the VDRL and RPR. Okay, we can also do specific treponemal antibody testing. Okay, so we have the treponemal tests. Okay, so we have VDRL or the Venereal Disease Research Laboratory test and RPR, or the rapid plasma reagent. These are non-treponemal tests. Okay, so VDRL, this test measures the antibody reagent in a patient who has formed antibody against cardiolipin, cholesterol, and lecithin. Okay, so this is involved with the principle of flocculation testing. Okay, so tests are read microscopically for the presence of flocculation. So reporting could be non-reactive, weak reactive, or reactive. So positive reaction with VDRL, okay, we can see microscopic clumps, clumps or microscopic flocculation. Okay, what we usually use, okay, is CSF, okay, but we can also use inactivated serum. Okay, we can use, okay, also inactivated serum. Okay, so reactivity during this disease, it may be negative in primary stage. Okay, titers usually peak during secondary or early late stages. So during treatment, okay, the reagent will become will decline, okay, and it will become non-reactive in the VDRL, especially 1 to 2 years following successful treatment. False positive can be caused by biological causes, okay, such as infectious mononucleosis, okay, conditions such as infectious mononucleosis, infectious hepatitis, malaria, leprosy, lupus erythematosus, rheumatoid arthritis, advanced aging, and pregnancy. Okay, reactive in other treponemal infections also seen in Yos and Pinta. Okay, this is also similar to the RPR. Okay, and we have RPR. Okay, RPR also in the principle of flocculation, detecting the reagent. Okay, we use charcoal here. Okay, we use charcoal. Okay, the charcoal is trapped in the flocculation reaction, which allows reaction to be seen macroscopically. So here we can see a macroscopic agglutination. Okay, so we use serum. We don't require, okay, inactivation of serum here. Okay, you can also use plasma. Okay, we can also use plasma. Okay, so this two assays are used for screening. Okay, and treatment monitoring. Okay, this is helpful in screening and treatment, okay, monitoring. Okay, so to confirm the presence of syphilis, okay, we should proceed with the treponemal test for syphilis, and this can be achieved using the fluorescent treponemal antibody absorption, or FTA, okay, ABS, treponemal pallidum particle agglutination, or your TPPA, and we have antibody capture enzyme linked immunosorbent assay, or ELISA. Okay, so sa tanong sa atin, ang ginamit natin is microhemagglutination assay, MHATP. Okay, this is also used to confirm, okay, syphilis infection. Okay, this is also used to confirm, okay, syphilis infection. For treponemal test, okay, we can use this to detect antibody to T pallidum. Okay, this can be used to detect, okay, antibodies to T pallidum. Okay, so we have fluorescent treponemal antibody absorption, which uses the principle of fluorescence. Okay, so here we have sorbent, okay, slides with Nichols chain of T pallidum, fluorescein labeled anti-human globulin. Okay, so positive reaction would present fluorescence. What we can use here as specimens are serum and CSF. Okay, and remember in FTA-ABS, it is usually positive before the reagent test. Okay, so reactivity during disease, okay, in FTAABS positive testing, okay, this is usually positive, okay, before reagent testing. Some false negatives in primary syphilis can also occur, okay, and it is usually positive for life. We have also treponema pallidum particle agglutination. Okay, here, okay, we have reagent colored gelatin particles coated with treponemal antigen. Okay? So, treponemal antigen is combined with liposomes. Okay? Your treponemal antigen is combined with liposomes. If antibodies are present, a mat of agglutination forms in well of a microtiter plate. Okay? So, positive reaction would be agglutination of sensitized gel particles. Okay? A smooth mat, okay, over surface of well would be observed. Okay, for here, CPPA, okay, the specimen could be serum. Okay, so this is not as sensitive as the FTA. Okay, sensitivity is close, although sensitivity is close to 100 in secondary syphilis. Okay, usually positive in late stages. Okay, itong TPPA. Antibody capture enzyme linked immunosorbent assay, okay, or ELISA. So, we have the enzyme labeled treponemal antigen that will capture the antibody T pallidum to T pallidum in the patient's serum. Okay, so what will be the positive reaction? A color development following the addition of the substrate. Okay, so here, okay, it presents a high sensitivity in primary syphilis. Okay, in primary syphilis, and it decreases in the later stages of the infection. Okay? So, in our question, okay, we use the MHATP. MHATP, sabi natin kanina, this is a micro heme agglutination test for T pallidum. Okay? So, MHATP. So, serum is mixed with sensitized RBC, okay, and the reactive reaction would present agglutination. Okay, would present, okay, agglutination. So, here, non-reactive siya, okay, and then the non-treponemal tests are reactive, okay, and this may suggest, okay, okay, a false positive. So, letter D is the correct answer. So, number 59, bullseye appearance rash is usually seen during what stage of Lyme disease? Okay, so remember, ang Lyme disease po, this is caused by an infection by Borrelia burgdorferi. Okay, Borrelia burgdorferi. Okay, so this is transmitted via saliva from the bite of tick. Okay? So, carrier ng Borrelia burgdorferi are your ticks. Okay? In ticks, okay, we have the Ixodes, okay, species. Okay? So, this infection can actually be prevented if the tick is removed within 24 to 34 hours from the host. And we have different stages of Lyme disease. Okay, namely, we have the early stage, okay, the late stage, the of Lyme disease. During the early stage of the Lyme disease, what we can observe, okay, are redness, okay, redness in the area, okay, 2 to 32 days after bite. Okay, and what we can observe here, okay, is a bull's eye rash, bullseye appearance rash. We call this as the erythema chronicum migrans. Okay, so the appearance of bullseye rash, okay, this is the erythema chronicum migrans, which is observed during early stage of Lyme disease. In the late stage of Lyme disease, the most common symptoms of this late stage is the presence of arthritis, okay, affecting the knees, okay, the knees, we have the shoulders and elbows. Okay, and also in the late stage of Lyme disease, we can have CNS involvement. Okay, it can affect the central nervous system of the patient, causing aseptic meningitis. What else? Facial nerve palsy and encephalitis. Okay, and also what we can observe in late stage, we can have a heart involvement or cardiac involvement. Okay, okay, so those are the significant presentation of stages of Lyme disease. So presence of bullseye appearance rash, okay, is seen in early stage of the Lyme disease. Okay, so diagnosis of um, Lyme disease, okay, this can be made in if a fourfold increase in titer, okay, is detected between an acute serum specimen and a specimen taken 6 to 4 weeks, okay, of infection. Okay, so what we can do, okay, a rapid method can be used, okay, in detecting IgM, okay, IgM antibodies to Burgdorferi, okay, antigens. Okay, so immunofluorescence and enzyme-linked immunosorbent assay can also be used, okay, as screening test. And we can be this can be confirmed using immunoblotting. Okay, so letter A is the correct answer. Number 60. What is the most common early abnormality observed, okay, observed in the lymph nodes of AIDS patient? Okay, what is the most common early abnormality observed in the lymph nodes of AIDS patient? Choices are letter A, reactive lymphadenopathy. Letter B, mantle cell lymphoma. Letter C, classic type of Hodgkin's lymphoma. Or letter B, lymphoblastic leukemia with T or translocation 922. So, the answer in number 60 is actually letter A, reactive lymphadenopathy. So, lymphadenopathy syndrome, this has been used to describe the first symptomatic stage of HIV progression preceding a diagnosis of AIDS. Okay, so generalized lymphadenopathy is the early sign of infection, okay, with human immunodeficiency virus. So follicular hyperplasia, okay, is the most common form of reactive lymphadenopathies occurring in infections such as HIV infection. So mantle cell lymphoma, this is not associated with HIV infection, but mantle cell lymphoma, this belongs to group of diseases known as non-Hodgkin's lymphoma, which are related malignancy that affect the lymphatic system. Okay? We have chronic Hodgkin lymphoma, a type of lymphoma where Reed-Sternberg cells are seen, okay, but this is not associated with HIV. Okay, we have the B lymphoblastic leukemia with translocation 922. Okay, so this is a Philadelphia positive ALL and not associated with HIV infection. So number 60 po is letter A. Okay, letter A. 61. AIDS patient have an increased susceptibility to infection due to a disease, due to a decrease in which of the following options? A, CD4 positive lymphocyte, B, CD8 positive lymphocyte, C, HIV antibodies, or letter D, HIV antigen. So, HIV, okay, it causes the acquired immunodeficiency syndrome. Okay, so the virus binds to the CD4 molecule on the T helper cells. Okay, so they have a specificity with the CD4 molecule of the T helper cell, but they can also attach or invade monocytes, macrophage, and other cells. Okay, so remember HIV, the virus, they have a primary target, which are the T helper cells, which are CD4 positive cells. So primary target nila si T helper cell. Okay, so it is the CD4 positive cells, the T lymphocytes, that help aid our immune system in attacking foreign antigen that cause infection. So HIV, they mainly targets these cells by using the virus glycoprotein to bind to the lymphocyte receptor. So once the virus is integrated into the cell, it begins to replicate the viral genome. Okay, so as viral replication progresses, the CD4 positive cell decrease, and leading to the increase of susceptibility to infection. Okay, so the answer for 61 is CD4 positive lymphocyte. Okay, so remember, pag lumabas dito, once the HIV, the newly transcribed HIV virus, buds out from the cell, eventually this CD4 positive cells will eventually be killed. CD8 positive cells also help in or has a role in immunity by eliminating cancer cells. So a decrease in these cells could indicate autoimmune disease but does not indicate an infection to HIV. HIV antibodies would not leave the host more susceptible to infection. So remember, the antibodies are not the only defense the body has. The antibody has generally stay stable during infection, but after antiretroviral therapy, the virus decreased, as well as the HIV antibody. Once the patient undergoes ART, the viral load will decrease, as so with the HIV antibody. HIV antigens are part of the virus itself. So therefore, a decrease in HIV antigens could mean a decrease in viral load. So 61 po, the answer is CD4 positive lymphocyte. So number 62, tests to identify infection with HIV fall into which three general classification types of tests? So we have letter A, tissue culture, antigen and antibody test. B, test for antigen, antibody, and nucleic acid. C, DNA probe, DNA amplification, and Western blot assay. Or letter D, ELISA, Western blot, and Southern blot. So two common methods for detecting antibodies of HIV, should be, this is detected by ELISA and Western blot test. We have also two common methods in detecting HIV antigens. HIV antigens, we have ELISA and also immunofluorescence. So we have also two common methods in detecting HIV genes. Okay? Kapag HIV genes, we can use southern blot test. Okay, we have southern blot test. Okay? We can also use DNA amplification or PCR. Okay, so DNA amplification. Okay, so DNA amplification using a PCR or polymerase chain reaction. These are used to detect viral nucleic acid in infected lymphocytes. Okay, so number 62, so pwedeng antigen, pwedeng antibody and HIV genes or nucleic acid test. So letter B is the correct answer. So remember, ELISA can detect antigens and antibodies to HIV. This can be the screening test for HIV. This can be used as a screening test for HIV. Okay. Other tests includes a Western blot, which is based on the recognition of the major HIV proteins by fractionating them according to their weight by electrophoresis, and then visualizing their binding patterns with specific antibodies. Okay, so Western blot appears to work best in samples containing high levels of antibody. And in Western blot, the presence of two of three bands, two of three bands must appear for a Western blot to be considered as positive. These bands could be the band binding pattern for P24, for GP41, GP120, or GP160. Okay? Other tests include RT-PCR. Okay, this is a RNA virus amplification method. Okay, and then indirect immunofluorescence. Okay, this can be used to detect HIV antigen in infected cells. Okay, so number 62 po, the answer is letter B. Okay, number 63. The major structural protein core of HIV virus is A. GP41, B. P24, okay, B. PE24, C. GP34, or letter D. GP140. So, HIV infection, this is commonly caused by HIV-1 virus. Although there are two types of HIV virus infecting humans, HIV-1 and HIV-2. So, commonly encountered sa HIV-1. Okay, so in infection to HIV, is confirmed using Western blot, the traditional confirmatory test for HIV. Although this is not as sensitive as the fourth generation EIA or enzyme immunoassay, and also by the nucleic acid amplification testing or NAAT, so interpretation of Western blot is most controversial among laboratories, but they report positive tests if at least two of the following three bands are present: P24, GP41, GP120, or GP160. Okay? Nucleic acid amplification testing required following negative or an intermediate result to Western blot. Okay, so kung negative ang or intermediate result tayo kay Western blot, what we can do is to do NAAT. Okay, so downside lang ni Western blot is it is time consuming and it is difficult to interpret. Okay, so other tests we can use indirect immunofluorescent assay or IFA, which also detects antibody to HIV. Okay, specificity and sensitivity of this test is comparable to Western blot. Okay, although this is not frequently used because or due to the expensive characteristic, and also it is prone to subjective interpretation. NAAT, on the other hand, detects HIV RNA. It is a qualitative test used for HIV infection confirmation. Okay, so here the major structural protein core of HIV is actually the P24 antigen. Okay, P24 antigen. So this is a core code for nucleic acids, detectable in 2 to 3 weeks Okay, and may become undetectable as the antibodies develop against it. Okay, then detectable again in late stages as immune system fails and the virus replicates, okay, in the body. Okay, so the answer for number 63 is P24. Okay, P24. Number 64, interpret the following results for HIV infection. ELISA, positive. Repeat ELISA, negative. Western blot, no bands. So these results are not indicative of HIV infection. Okay, not indicative of HIV infection. Okay, maybe due to a testing error in the first ELISA, okay, testing. Okay, known false positive ELISA reaction occur in autoimmune. Okay, false positive reaction to ELISA, caused by autoimmune disorders. What else? Syphilis, alcoholism, um, lymphoproliferative disease. Okay, so these are the common causes of false positive, okay, results to HIV ELISA. Okay, the presence of autoimmune diseases, syphilis, alcoholism, and lymphoproliferative diseases. So a sample is considered positive if HIV is repeatedly positive by ELISA or other screening method and positive by a confirmatory method. So in this case, okay, this is a negative test for or negative assay for HIV. In number 65, what molecular techniques is appropriate for HIV-1 genotyping? A. Reverse transcriptase polymerase chain reaction. B. Multiplex polymerase chain reaction. C. Real-time polymerase chain reaction, or letter D. Strand displacement amplification. So remember, we do amplification testing. So sa HIV, we detect HIV RNA. Tama ba ako? So HIV RNA ito. So we use reverse transcriptase polymerase chain reaction for this. So RT-PCR. Okay, RT-PCR. So, this is a procedure that can be modified to include the conversion of RNA to DNA using reverse transcriptase in the initial steps. Okay, so this is helpful in detecting the viral, okay, RNA, okay, viral RNA agents such as here, human immunodeficiency virus. In particular, we have the HIV-1 genotyping. Okay, ang multiplex polymerase chain reaction, this is used, okay, in this uses, kaya siya sinabi multiplex, okay, it uses multiple primers, okay, in a single reaction tube. So ibig sabihin, kunwari, in one reaction tube, in one Effendorf, okay, yun yung ginagamit na parang test tube sa ating amplification testing. Okay, sa ating Effendorf, marami tayong primers doon, okay, and once run in a single reaction, we can, okay, target different, okay, fragments of nucleic acid. Okay, so specific nucleic acid amplification should occur if the appropriate target, actually usually DNA, okay, is present in the sample test. Okay, that is used in multiple or identified in multiplex polymerase chain reaction. While in real-time polymerase chain reaction, okay, real-time polymerase chain reaction, actually, para lang din siyang the conventional polymerase chain reaction, but in this case, okay, it uses the fluorescence resonance energy. Okay, fluorescence resonance energy. Okay, so this is used to transfer, okay, signals to quantitate specific DNA sequences of interest and to identify point of mutation. So habang nandun pa siya kay machine, we can see the rise of amplicons or amplified nucleic acid. Okay, kahit pa nandun pa siya sa ating machine, PCR machine. Okay? So, unlike other tests, other PCR, okay, in real-time polymerase chain reaction, habang nag-amplify tayo, nakikita natin na nagpi-pick up siya. Okay, unlike sa conventional PCR, at the end of the PCR, okay, of the cycles, that's the time we're going to test for the presence of amplified nucleic acid. We have strand displacement amplification. This is a method that amplifies target nucleic acid without the use of thermocycler. So thermocycler is the PCR machine. Okay, so here in strand displacement amplification, okay, we amplify nucleic acid without the use of that PCR machine or thermocycler. So a double-stranded DNA fragment is created and becomes the target exponential amplification, and it is not used usually in HIV testing. Okay, so gumagamit tayo dito ng DNA fragment. Okay, so here, in number 65, the answer po is letter A, reverse transcriptase polymerase chain reaction. Okay? So, we need the reverse transcriptase, okay, in order to convert RNA to DNA, and that will be tested or amplified later on, okay, in the thermocycler. Okay? So, that is number 65. So again po, salamat po and good luck po in your examination. Hopefully, we were able to help you study, okay, in despite of the suspension of classes, okay, because of the COVID-19. Stay healthy and stay safe po, ma'am and sir.